Meiosis is the process by which an organism's identical diploid germ cells are split into 4 non-identical haploid gametes. The law of segregation states that each gamete contains only one allele from the parent cell. Mendel's law of independent assortment states that the genes are independently sorted from the parent into gametes. The cell begins with interphase. Interphase before meiosis is the same as interphase before mitosis. During the G1 phase, the cell grows until it becomes the desired size. In the S phase, the chromosomes are replicated, the chromatin is uncondensed, and the centrosome is duplicated. In G2, the cell prepares for meiosis. The next step is prophase 1, the longest stage of meiosis. During this stage, chromosomes begin to condense. Synapsis occurs where chromosomes begin to pair up with their homologs to form homologous pairs. The homologous pairs are made of one chromosome from each pair. As they align genes with each other, crossing over occurs, a process by which segments of each chromosome are exchanged with the other. Chiasmata are the X-shaped regions where crossing over occurs. The chromosomes then form tetrads, bundles of 2 homologous chromosomes, which each consist of 2 sister chromatids. As the nuclear envelope begins to fragment, the spindle microtubules begin to form. The kinetochores attach to the microtubules, allowing the homologous tetrads to move to the metaphase plates. After that, metaphase 1 begins. The homologous tetrads line up along the metaphase plates, an imaginary line where the cell will bisect. Microtubules form the centrosomes, extend and pinch themselves at the kinetochores of homologous chromosomes. According to Mendel's law of independent assortment, the homologous chromosomes line up on the metaphase plates in no particular order and are very random. Thus, the sorting of one allele of a gene into gametes does not influence the sorting of another allele into gametes for a different gene. During anaphase 1, the tetrad splits into 2 homologous chromosomes, which are pulled to opposite poles of the cell. Kinetochores attached at the centromere of the homologous chromosomes pull the chromosomes towards the opposite poles by the shortening of the microtubules of the spindle apparatus. The shortening is caused by the kinetochore depolymerizing the microtubules into tubulin subunits, and as the microtubules shorten, the chromosomes are pulled toward the centrosomes. The cell proceeds to telophase 1, where the spindle apparatus is used to pull apart the homologous chromosomes via the centromere, and then cytokinesis begins as well. In animal cells, this is a stage where the cleavage furrow forms, pinching the cell in two, and in plant cells, this is a stage where cell wall forms along the metaphase plate. The cell prepares for another stage of division. The daughter cells do not replicate their chromosomes before meiosis 2, which means that the products are haploid cells. Another round of dividing begins with prophase 2. A spindle apparatus begins to form around the cell, and the chromosomes line up along the center. Their kinetochores attach to microtubules, and the chromosomes move towards the metaphase 2 plate. At metaphase 2, the spindle apparatus is now attached to the centromeres in the chromosomes, which are lined up along the metaphase plate. Each chromosome consists of 2 non-genetically identical chromatids and 2 kinetochores attached to the centromere, one for each sister chromatid. Kinetochores are attached to the microtubules of opposite ends of the cell. During anaphase 2, the centromere separates and the sister chromatids are pulled to opposite poles by the shortening of the microtubules. The shortening is caused by the kinetochore depolymerizing the microtubules into tubulin subunits. As the microtubules shorten, the chromosomes are pulled towards the centrosomes. During telophase 2, the chromatin decondenses and 4 haploid gametes are formed, each being genetically distinct from one another. The haploid gametes end up as 4 sperm cells or 1 egg cell.